Good afternoon, children. Yes. Good afternoon. Shillong, Churachandpur, Itanagar. How are you? There's some interesting game going on at Shillong Center. I wish I could join you people. Okay. Fine. Okay. Settled, all of you? Had your lunch? Yeah, I know, sleepy weather. Same here. Me too, I am feeling very sleepy. Okay. Settled, everybody? Okay, don't sleep in my class. After the class, you can obviously sleep. But I wish you don't sleep in my class. Okay. Shillong, how are you? Fine? Yes, Shillong. Churachandpur. No, ma'am, never. Why? You're not sleepy? Okay, I could sleep in any of the classes. After lunch, obviously. Okay, fine. So we were discussing something important, isn't it? You had a good break, 30 minutes, isn't it? Okay, thank you, Itanagar. Okay, fine. So let's continue with the class. We were talking about the skeletal muscle system in the last class, where we on Wednesday, I guess, isn't it? So after that, we are going to obviously today see the actin and myosin, what is its basic structure, the ultrastructure of actin and myosin, and how the whole event of contraction of the muscles are going to take place, and that causes what? That causes the whole locomotion or movement in our system. So if you are writing, just suppose you are writing, so we will be considering these kinds of movements under voluntary movements or the movements called as skeletal movements. So we are talking about those muscles in this class which are attached to your bones, that is called as skeletal muscles, isn't it? So what is chapter coming in our test? I don't know. I need to ask your coordinators what are they planning actually. Let me ask once. I will be telling you that by tomorrow, or updating on the group itself. I will ask them to update on the group. Okay, so what have you finished in botany? I guess we have finished a good portion in zoology part, isn't it? So digestion is like you have given test. Breathing is like the next upcoming test can be of breathing. Can be. I'm not sure. I will be updating this on group today. Let me ask. Okay, fine. So we are going to continue with the same topic, actin and myosin. So last concept was clear to everybody, I guess, isn't it? So can I start with actin, the ultrastructure of actin? The whole scenario was clear, the H zone region, isn't it? The H zone, I band, A band, M line, isn't it? I'm not sure, child, I'm not sure. Shillong, I will be updating you by today evening itself. I will ask somebody to update that. What is the test syllabus? I'll just ask somebody to update on group. Okay, don't worry. By today itself, I will be asking to update. Okay? Don't worry about it. By today evening, I will be asking. Okay, fine. So let me continue with that particular thing which we're talking about. That was A band. Remember, A band was the dark band, isn't it? When we talk about A band, the A band was the dark band, the dark region, isn't it? The dark region was dark because it has overlapping of what? It had overlapping of both actin as well as myosin. And in between dark band, there was something. What was it? In between dark band, there was a light colored zone. What was it? Who is going to tell me? Churachandpur, Itanagar, Shillong, who is going to tell me? The light colored zone. The name of the zone, yes. Let me see who's going to answer me first. Yes, Churachandpur, Shillong, Itanagar. In between the dark band, what was the light colored zone? Yes. Yes, Shillong. No. Sarcomere was the whole functional unit. The basic unit of contraction was sarcomere. Just let's have a quick revision of the diagram. I guess we need to have a quick revision. See. Can you see this? All of you? Excuse me. See, let's see. This was the region, if you take an account that this region where was only actin, this was the I band. The region with only actin and myosin overlapping, this was the A band. Remember? And then we had a region where there was no myosin but only no actin but only myosin, and this region is what? This region is called as H zone. So H zone is what? H zone is the light colored band or light zone between the dark band. So in between the dark band you have a light colored region, and that region is called as H zone. Now similarly in between the I band you have a Z line. So I told in the last class also, Z line actually holds the I band on either sides. On either of the sides the Z line is holding the actin myofilament. So the Z line is what? This Z line is actually the dark region. This happens to be the dark region in between the I band. So you have a light colored zone called as I band, light band, that is I band, and in between I band you have a dark region called as Z line. So what was sarcomere? Remember, when you are talking about two Z lines, two consecutive Z lines like these two, this is also a Z line and this is also a Z line. So this distance between two Z lines is basically called as what? Is called as sarcomere. So sarcomere is nothing but the sarcomere is the basic unit of contraction in your muscle fiber. So whatever is contracting, that is actually sarcomere, the basic unit. So when there are like a group of sarcomeres contracting together, the whole contraction procedure, it happens. So if you are writing, suppose it's actually because of millions of sarcomeres, they are undergoing contraction at the same time. So this is what is the basic concept. Let's move ahead with the basic structure of actin filament. So I guess this must be clear. The I band contains only actin. If you can see over here, it has only actin. And if you talk about A band if you can see, it has both actin and myosin overlapping. So the region in which actin and myosin are overlapping is basically called as A band. Clear to one and all? Yes, children, tell me. Shillong, Itanagar. Churachandpur, where are you people? Yes, Churachandpur, where are you children? Are we connected? Please let me know. Okay, fine. Shillong, is this clear? And there was one more region in between this. Remember, there was this particular green thing which I am drawing. There was a region which holds the Miocene at its place, and we call this what? We call this to be M line. If you just flip your pages. Okay, Churachandpur, are you getting everything? Tell me, children, are you getting whatever is being taught? Okay, fine. So let's move ahead. So this is what would be the answer now? Tell me. Tell me the answer now. In between dark band there was a light colored zone. What was it? Yes, very good. So the answer is now, very good Shillong, the answer is now H zone, isn't it? This is what we just did now. This is H zone. Okay, fine. So in between light band there is a dark region. Name it. Itanagar, Shillong, come on. Name it. It's called as, dark region is called as what? Quick. Name this region. Shillong, Itanagar, Churachandpur. Quickly answer this. See, A band is never in between the light band. Read the question properly. Shillong, read the question properly. The A band is never between the light band. A band is a different unit. Light band or the I band is a different unit. And my question is that in between I band, what is the dark region? See, I have got a correct answer from Churachandpur. Very good. So this is the dark region in between the I band. So if suppose this is the whole scenario, okay, let me just highlight the whole I band scenario. So this is, suppose the yellow part, yellow section is I band. So the line running in between, this line, I am talking about this line running in between, this happens to be the dark region. And what is this dark region? This is called as Z line. So your answer should be what? Your answer should be Z line. So in between A band there is a dark, light region called A band or A zone, H zone, I'm sorry. And then in between the I band you have a dark region, and that dark region is called as Z line. Fine. So this should be your answer. So let's now have a look at the ultra structure of actin. So how is an actin? This is the basic structure of actin. Let's see quickly. So just draw the skeletal structure first. Okay. Just try to follow. So I am drawing one helix. This is one helix, and the same color because they are actually made up of some same subunits. So I need to have the same colors for the second helix as well. So you have two helix, and let's quickly line out the helix with a different color. This is the, so okay children, this is clear to one and all? Yes, tell me kids, this is clear? This green section over the blue balls must be. So this is the ultra structure of actin. So actin is what? Actin is a double helix. Now we need to put certain more structures over this. This is what is called as tropomyosin, and this small structure which I am drawing in pink is the troponin part. So this green part is tropomyosin, and this pink section on that, the pink structures on them is called as troponin. So this happens to be troponin, this green thing happens to be tropomyosin, okay, and this blue thing a single unit. If I talk about single structure, so if you know, do people know a kind of sweet dish called as laddu? Yes, tell me. It's very popular in North India. Do you know that? Laddu? All of you? Shillong, Itanagar, Chuachandpur. Have you heard about laddu? So this is a single laddu. This is called as G-actin. So G-actin is what? G-actin stands for globular actin. So a single laddu is, yes, it's tasty. Now I wish to eat it. I will be eating it when my class is over. So this is what is basically the single laddu part. The single laddu is called as G-actin. Okay, this is globular actin. So this is actually a monomer. And when you line together many laddus in a single line, okay, just take many laddus and just make a chain-like structure. The whole chain-like structure is called as F-actin. F stands for fibrous actin. Okay? Am I clear? So what you have to take? You have to take a single laddu. Suppose I am shading this single laddu in blue, and you will be taking such kind of many laddus and you are going to line them in two different rows in a helix, just like your DNA. Okay, and then the structure you will be getting is basically a single strand of fibrous actin. So in total you have over here how many fibrous actin? In total you have over here 2 fibrous actins. So your actin myofilament is made up of basically these 3 subunits. The first subunit being the F-actin, this is the first thing. Then second is having the tropomyosin, and third we are having is troponin. So these 3 things together, they compose your actin. So your actin myofilament is not a single unit, but it is composed of what? 1, 2 and 3. You know now what is 1. 1 is F-actin or fibrous actin. 2 is tropomyosin. And 3 is troponin. So you have actin composed of 3 subunits. This is quite important. These kind of questions, they do come in exam. Yes, Shillong. What is going on? Feeling very, very hot? Okay. It's hot over there, hot and humid, I guess. Is it? And what about the weather at Itanagar and Churachandpur? Is it hot? Okay. It's same in Delhi also. It's very hot, humid. Rather than hot, it's humid. Okay, fine. So you need an ion. This again a question comes over here. To polymerize this, you need magnesium ion. So a question often comes, what is the particular ion you require for the polymerization of G-actin into F-actin? Okay. Okay, Shillong children are not having AC, I guess. Okay. Fine. Okay, fine. So this is what is we are talking about till now. This is the ultra structure of actin. So if a question comes to you, what is actin made up of? You have to answer that actin is made up of number 1 F-actin, number 2 tropomyosin, and number 3 troponin. Now let's see the ultra structure of the 2 and 3. 1 is clear. What was 1? 1 was fibrous actin. So what we do is that we have 2 helical strands for fibrous actin, and each strand of fibrous actin is made up of what? When you talk about each strand of fibrous actin, yes, each strand of F-actin or fibrous actin is made up of monomers of, remember laddus, monomers of G-actin. And what is this G-actin? This G-actin is nothing but it is globular actin. So we talk about this thing, this is how you have to explain the first part. That is F-actin section. So actually speaking, F-actin is actually a polymer. And for the purpose of polymerization, that is when you convert the G-actin to F-actin, what I need is, I need a magnesium ion for the same. So this is a particular activity you need to have. This is quite important. You need to learn for your NEET as well. This is very, very important. Just let me know if things are not clear. Centers. Yes, Chuachampur, Intanagar, Shillong. Tell me children if anything is not clear up till now. The first section is complete. Yes, Shillong. Yes, children, what is happening? Any problem? Shillong, tell me. Is there any problem? Yes. Okay, fine. So I'm moving ahead. I guess you must have copied this slide, all of you. Did you copy this slide? Okay, fine. I'm moving ahead. Okay, Shillong seems to be very busy today. What is the issue, Shillong? You people are quite busy today. Now we have is tropomyosin. Now tropomyosin is the second subunit. This is made up of 3 polypeptide chains. That are wind together. So we need to have 3 chains of polypeptide, and what we are doing is that a simple kind of structure that you use to make your egg pony. Isn't it? The girls will understand this thing very nicely. The way you actually weave your hair, making egg pony, that is 3 strands wound into a single structure to form a tropomyosin subunit. And number 3 was what? Number 3 was troponin. Now this troponin is a trimeric protein. What is it? It is a trimeric protein. Trimeric means it has 3 subunits. Okay, so it is a trimeric protein. As you can see the structure over here also, we made 3 circles one over other. Isn't it? So similar kind of structure is followed in troponin. What is troponin? Troponin is a trimeric structure. One of the subunits binds to calcium. one binds to tropomyosin and one binds to the G-actin. So this is the whole summary of the actin, how they bind together, how they are going to work together. Done? Up to this? All clear? Let me know if any issues. Yes, children? Just let me know if any issues. Shillong, you seem to be very inert today. What is the issue? Yes, Shillong? All clear? Are people able to chat today? Yes, I guess you are. Yes, Shillong? Fine. Okay, don't be crossed here and there. Please be attentive in the class. This is a quite important chapter. So we are having what? We are having 3 subunits in troponin, and what are they? One of them binds to calcium. One binds to tropomyosin. Excuse me. And one binds to G-actin. Let's see. Now, the next scenario. This was all about the actin part. Now let's start with myosin. Let's see about myosin. Now the myosin is actually again I don't know what is happening. Just wait for a minute. Let me draw it properly. You can copy after that. I guess today is not my day for drawing. Okay, fine. Yes. So we have the tail regions of these structures wound together. So the myosin again has 2 parts. This unit is called as LMM, as in light meromyosin. And these 2 subunits are called as the head region or the HMM. So they are called the heavy meromyosin. So meromyosin is the subunit of the myosin, and they are having 2 substructures or 2 divisions, better say, the HMM part and the LMM part. HMM stands for heavy meromyosin and LMM stands for light meromyosin. And then we have 2 basic sites over here. Let me just mark for you. You have a pink section over here and you have a, let's say, a green part over here. So this green section is actually the particular region to which ATP binds, and it has an enzyme called as ATPase. Now we are at a particular position. We now understand that if you talk about an enzyme, how do you nomenclate it, how do you name it? You just put ASE after the substrate. So if I am talking about ATPase, obviously it's an enzyme that breaks down ATP. So an ATP breaking down enzyme would be called as ATPase, and this is a site where ATP binds. So let's mark over here also that this is a site where ATP binds. Quite important. So ATP would be bound to this site, and after its binding, what happens is they undergo breakdown. And these two pink regions are actin binding sites. These two pink zones are what? They are the actin binding sites. So this is the particular kind of configuration we look into when talking about actin and myosin. Actin was a little complex compared to myosin, I know, but obviously both of them are going to form a cross bridge. I will show you just now how. Just let me know if you are done. So this is actin binding site on the myosin. So myosin would be having actin binding site and actin would be having myosin binding site. Am I clear? So actin has a myosin binding site and myosin has an actin binding site, so that both can associate with each other, both can bind with each other and form a cross bridge. Okay, fine. Are we clear up to this? Itanagar, Shillong, Chuachampur? Yes. Shillong, give me a thumbs up today. Today people are very busy. It seems so. Yes. Shillong? Okay. Just give me a thumbs up. Did you get a punch? The girl on the second bench? Did you get a punch on your face? Yes. Oh, are you hurt? Are you fine, child? Are you okay? Okay, fine. Okay, all of you, just give me a thumbs up if you are getting whatever is being taught. Don't do that dhishum in the class. You people might get hurt. Isn't it? Don't do that. Okay. All of you are clear? Just give me a thumbs up, Shillong. Chuachampur, tell me, Itanagar, tell me if we can proceed ahead. Yes. Okay. Fine. Very good. So this is what is the particular kind of association I am talking about. Now let's see the particular structure, how it is supposed. So this was what? This was the actin. Now actin was what kind of structure? Actin was a helix of this kind, isn't it? It was a helix of this kind. Remember, just now we have done this thing. So there are many laddus and each laddu is lined in a line to form a fibrous actin. So this was kind of structure we are having. This whole thing is very bad. I guess today is not my day for drawing. Okay, let's retry it. Let's draw the laddus in pink. So there are many laddus lined together again. So this is what is the actin. This is what is the actin particular thing. So there are particular strands of actin, and between those strands what we have is we have myosin. So let's see myosin first. So how is myosin? Myosin is actually somewhat this kind of structure. See this. Okay, and there are this kind of loops arising. This is what is the head part. So head part of the myosin is arising. I told you today is not my day for drawing. I won't be able to draw, I guess, today. Okay, this is what is the head part. Yes, finally. This is what is the head part. So both of them would be inclined in a different direction. If I am talking about this region, this is what is the typical structure we are going to have. Yes, I am somewhat able to draw something. Again the pink laddus. Yes, finally. So what happens is this particular diagram, it actually shows a whole kind of scenario which is present in this A band and I band, that is between the A band. So what happens is this particular structure, that is the myosin, it forms a cross bridge with the actin. Just see the yellow section I am trying to draw. this whole thing, this myosin goes and binds to the actin, and actin obviously binds to the myosin. Look over here everybody, and what happens is this section of Z line starts to get towards each other. So what happens is, as soon as the myosin, just imagine these are the two myosin strands, okay. Just imagine yourself to be in the center of the class and you are having two ropes in your either hands and you're just trying to pull the whole wall section. The walls on the either side are the Z lines, okay. The walls on the both the sides are Z lines and you are having two ropes in your hands, one in each. And then you are trying to, this is a particular structure the myosin is having. Just look at your diagram you have drawn. We have drawn a particular kind of structure which is having many myosin like this. So what is going to happen, every time an actin is going to attach, and finally that actin is attached to the walls of your classroom, both the walls on the either side. And what we are going to do is that we are going to pull the walls like this, okay. So what would happen, finally the walls get pulled and the sarcomere length or the Z lines, they come closer to each other. So this whole process is said to be the muscle has contracted. Let's see once again what happens. The myosin, it is somewhat like this in structure. Just look at my wrist position. Both of them are like this in position. And then what happens is they start to swirl. They start to rotate, and in a way that they are going to pull both the walls towards each other. Okay, let's see in the diagram once again what is going to happen. This is going to pull the walls towards each other. So my sarcomere length is going to decrease after this. This is going to be pulled in this way and this is going to be pulled in this way. Okay, so they are going to be pulled in this way, and the particular pulling will be to that moment until this whole thing becomes zero. That is until the actins they collide with each other. The whole scenario would be repeated. So the particular pulling section will happen, and once the actin is totally merged with each other from both the sides, what we would be having is that we would say that yes, the muscle contraction has taken place, it has been done. Am I clear? All of you? All of you, am I clear? So what is going to happen over here? So this is what is actin. Now this actin is actually going to form a cross bridge with myosin. See, in the previous structure I showed you myosin was not bound to it, but yes, myosin is now going to bind to this section. So actin must be having a unit. Actin has a particular structure. This is called the myosin binding site. And the myosin also has a structure. What is it? This is called the actin binding site. So we have these structures. This is what? This is a laddu, the G-actin. So all the G-actin, all the particular monomers, they have this particular binding site. And what is this? This is the single meromyosin unit. Remember, many units of meromyosin together they form the myosin or the heavy myofibril. So what they are going to do is that they are going to associate together, and this association will form what? This is going to form the actin myosin cross bridge. So once this cross bridge is formed, once we are done with this cross bridge thing, once the cross bridge is formed, now the actin and myosin start to move towards each other. So once you are done with the formation of actin and myosin, see, actin and myosin cross bridge is formed, and what happens is the actin along with the Z line is started to pull towards each other. This is the contraction theory. This is how the contraction works. But yes, how would this happen? Why would this happen? Why would this cross bridge be formed? Why? Since now you must be aware that this particular binding must be requiring a stimulus, isn't it? It requires a stimulus. Have you copied this? Can I move ahead? Itanagar, Churachandpur, Shillong. Have you copied this, children? No? Okay, copy please. Okay. Shillong, have you copied? Okay, very good. So I'm moving ahead. So what is happening over here? So I talked about troponin. See, we talked about troponin, and one of the troponin unit binds to G-actin. You have copied this, isn't it? So the troponin is actually masking the site. This is my actin. My troponin is present over here. Let me draw this in different color. My troponin is present over here in a way that it is masking the site. This is one of the units of troponin present here. I am having the second unit of troponin. See this, the next subunit. This binds to tropomyosin, and this binds to G-actin. Where to G-actin? This binds to G-actin at myosin binding site. So we could say that this particular unit masks the G-actin myosin binding site and makes its binding to myosin impossible, isn't it? So until and unless you are going to remove this particular part, this I am mentioning in green, let me draw once again. Unless and until you are removing this green section or the green part, you are not going to have this kind of bridge formation that just now you saw, this spine part thing. You won't be able to have this thing. So what we need to do is that we need to first of all remove this tropomyosin, and tropomyosin was having one more section, if you remember. Let me draw this in blue. So it was having one more section which is actually the calcium binding unit, remember? So let's see what actually happens, the whole scenario, what is going to happen. Let's have a flow chart quickly. So this subunit binds to calcium, but yes, at this particular point it is not binding to calcium. Let's see why. So this makes the whole story, this is a good concept. So G-actin is what? G-actin is actually masked, or you can say the myosin binding site of the G-actin is masked by the troponin in a way that there is no cross bridge formation. And one of these subunits binds to tropomyosin as we have mentioned, and the third subunit binds to calcium, and this also we have mentioned earlier. Okay, fine children, can I move ahead? Is this section clear? Shillong, Churachandpur, Itanagar, are we clear up to this? So whole scenario starts with a stimulus. We need to have a stimulus, and when many stimuli come together, they are called as stimuli. So there is a stimuli, and because of this stimuli what happens is the calcium ion from the sarcoplasmic reticulum starts to diffuse out. So what is sarcoplasmic reticulum? Sarcoplasmic reticulum is nothing but it is the endoplasmic reticulum of a muscle cell. So when you're talking about muscle cells, the endoplasmic reticulum of a muscle cell is called as sarcoplasmic reticulum. So calcium from the sarcoplasmic reticulum starts to diffuse out, number one thing. Once it is starting to diffuse out, what happens is this calcium binds to troponin. Obviously, we have just now studied that troponin has a calcium binding subunit. Remember? Just now I have told you. So, calcium binds to troponin over that subunit. Now, as soon as the calcium binding of calcium to troponin makes the troponin to change its orientation. So it changes its orientation. How? In a way that the myosin binding site on actin is free, isn't it? How? In a way that the myosin binding site on the actin is now free. So you are having what now? You are having a free site on the myosin. Okay, the myosin binding site on the actin is now free. Fine? Am I clear up to this? So the masking is removed. This thing actually will be free now. So the structure actually is somewhat changed. The orientation changes in such a way that if we talk about this section, this was the section, the green thing has been removed from its surface somewhat like this. There is a green part, the red part and the blue part. So what happens is this masking is now free. They are now free to bind to myosin. It can now freely bind to myosin. Yes. Am I clear? Can you see this? This whole thing has changed its orientation. Orientation means with respect to three-dimensional structure, its whole configuration has changed. So suppose it was like this, now it has configured itself to be like this. So it has changed its position with respect to the three-dimensional space. What we call in chemistry, that was stereoscopic. Fine? So this is the whole scenario. Let's see once again quickly. A stimuli comes. The stimuli causes the calcium from sarcoplasmic reticulum to diffuse out, and once the calcium starts to diffuse out, it binds to troponin section, troponin part. Which part? The calcium binding subunit part, and this calcium binding subunit to the troponin makes the troponin whole structure. Obviously this is very, very legible. Makes the troponin to change its orientation. So whole orientation is going to change in a way that the myosin binding site on actin is now free. So now actin is ready for binding to myosin. Now there are few changes in the myosin also. Let's see what. So once this thing happens, what would happen? Myosin has ATP bound. So what would happen when there was a stimuli? This ATP breaks down. The bound ATP breaks down. Breaks down to form what? We know this is ATP, and this breaks by the help of enzyme ATPase into ADP plus pi plus energy. Clear? So what happens? This is the next section we are having. So simultaneously two things would be running. The first was whole scenario about actin, and how troponin was actually covering its myosin binding site, and now how it has uncovered it. Now myosin has what? Myosin has ATP bound to it, and once the stimuli comes, the ATP breaks down. The breaking down of ATP releases ATP plus ADP plus pi and this energy. Now what is the whole use of breaking down ATP? Since like starting of the classes, we know now by now that whenever we break down ATP, we are interested in the energy section. So something is going to happen with this energy. Let's see what. So this energy is used to, the energy from ATP is used to form a cross bridge between actin and myosin. Is used to form a cross bridge between actin and myosin, isn't it? As well as two things, as well as to rotate the head of myosin. So the myosin head is also rotated. Let's see how the diagram. So this is somewhat your actin binding to myosin. This whole section is somewhat like this. I just now shown. So this is the cross bridge what is going to form after ATP comes. After ATP comes, and once the ATP is hydrolyzed, this cross bridge would be formed. So this is what is called as actin myosin cross bridge, and this requires what? This requires obviously energy, huge amount of energy. So now what is going to happen? This head is going to swirl somewhat like this. This is going to rotate. Remember I showed you that thing with the help of my fist just now. Just now I was showing you that physically by example, or giving example of the wall. So here we are. So if this was actin now, this was actin, this particular position, obviously it has shown a displacement towards inner side. So this is what is the actin myosin cross bridge formation significant. See, can you see this? It has pulled it towards its inner side. So if this was a Z line, obviously this was a Z line. So Z line has started to move. Z line has started to move towards the inner side. Why? Because we have actually broken down ATP, and this ATP is utilized to first form the cross bridge. This is what is the ATP two-way action. The cross bridge is formed. This is the actin myosin cross bridge. Okay, children, and this is the rotation of the head, rotation of the myosin head. So once it binds to the myosin, suppose this is the myosin section, it has bound to it. This is the actin part, this whole thing is actin. Let's take some other example. Are you taking this thing? Okay, my phone. So this is what is the actin, just see this thing, and this is the myosin part. So if myosin is somewhat like this now, just see the first part when cross bridge is formed, and then what it does is that after cross bridge formation swells its head. So swelling head means it becomes somewhat like this and the actin part is pulled. So this actually makes the whole section of the Z line to come closer to each other. So this is the depiction. So it rotates the head of the myosin, forms cross bridge first and then rotates the head of the myosin, and as a result of which what happens, just let me know if you don't copy anything, just don't miss out something, this is quite important. So once the head part is rotated, what would happen? The actin filament is pulled inward, or we could say what, Z lines come closer to each other, and we say what? We say that yes, the muscle contraction has took place. Now the muscle is said to be contracted. So this is the whole contraction process. We're done with contraction part. So contraction section is done now. We are having a muscle which is now contracted. We can say yes, the muscle has contracted now. So let's once again see this thing. So first of all, I need a stimulus. Many stimuli, in fact, it needs to be of threshold value. Threshold stimulus, you know what is it? It's all or none law of all over the muscle. Either the contraction will take place or not take place. Just like physics, you must have studied all or none law till now in physics. Okay. So you have calcium from the sarcoplasmic reticulum. It starts to diffuse out first, and this calcium binds to troponin. The calcium binding subunit of the troponin, obviously, this binding of the troponin makes the troponin to change its orientation, and in the similar way, the myosin also starts to change. How? That stimulus breaks down the ATP into ADP plus pi plus energy. Clear? Now this particular energy is used to do two things. What are they? First, to form a cross bridge, and second, to swivel or to rotate the head of the myosin. That is a heavy myosin. So once this happens, the muscle is said to be contracted. Now I need my muscle to be relaxed. So what happens is, because until and unless the muscle is relaxed, it won't be ready for the next stimulus. So what we need an alternation of contraction, relaxation, contraction, relaxation. So this is what is the particular way your muscle should move. So after every contraction, a relaxation is a must. Otherwise, the muscle will be set to be in twitch. You must have studied what is twitch or spasm. This is what is a twitch or spasm, when the muscle fails to regain its relaxed state after contraction. So after contraction, what I need to make my muscle relaxed. So for relaxation, what happens is, first and foremost, the cross bridge breaks, because the cross bridge was formed at only one condition, that I need ATP. Once the ATP energy is utilized, obviously cross bridge would break. Once the cross bridge breaks, what would happen? The myosin head resumes its original position. So it will be again back to its position, and what we need now is the calcium ion. Calcium moves from troponin, so it is going to dissociate from the troponin into sarcoplasmic reticulum. So there are often questions asked that this is an active process or passive process. The first, when you are like removing out calcium, was passive, because it was along the concentration gradient, because sarcoplasmic reticulum is a reservoir of calcium. It has many calcium ions. So at that point of time, in the initial period, it was a passive process, because from higher concentration to lower concentration. But now you need to push it back from a lower concentration to again a higher concentration. So now you will be having a active process. So you need to apply pump. So calcium moves from troponin to sarcoplasmic reticulum, and finally the myosin head binds an ATP. So these are two important things that are obviously that are a must. That is, number one, it should bind an ATP, and number two, calcium should move away from the sarcoplasmic into the sarcoplasmic reticulum. So these are two important things that should take place whenever we talk about relaxation of the muscle. Am I clear? All of you, if this thing does not happen, if the particular person or the particular person is said to be, not exactly person, this happens in dead bodies and this is said to be rigor mortis. You must have heard about it. Just let me know if the color is too dark. Shall I just, okay. I guess it's very dark to see through it. Okay, fine. So let's just underline it. Or I will be using another color. This is better. Okay, fine. So these are two things. For again a contraction to take place, I will be breaking down this ATP this time. Again, if I need to contract, I will be breaking this ATP, and that ATP is going to form a cross bridge. Once the cross bridge is formed, what would happen? The swirling of head will happen of the myosin, and once the swirling of head has been done, the muscle will be set to be in contraction position. So am I clear up to this? Let's see a small video regarding this in case you have any confusion. In the meantime, you can tell me about it also. Yes, Shillong, are we clear up to this? Sorted everything? Yes, Shillong, Chua Champur, everything is sorted. Let's see a small video.
Smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T-tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes. You use muscles every day to do activities.
Okay, was something clear? Was it clear? Tell me. Wish to see the video again? Or it was clear? So the whole process is very much clear with this video. So one thing is very much clear, that yes, the contraction also needs a ATP and ATP, and the relaxation is also initiated by ATP. So this whole process of relaxation will also start when an ATP binds to myosin. So when myosin is bound by an ATP, then only it is going to free the cross bridge. And this, if absent, if it is absent, this particular ATP molecule, if absent, the muscle will be in a contraction state, and what we say is, particular condition is said to be rigor mortis. You must have studied that thing that happens in dead bodies. The dead bodies gradually becomes stiff, very stiff. So what is going on, Shillong? What happened? Yes? What is it? Yes? We have a Shah Rukh Khan in class. Yes, Shillong? What is going on? Tell me. Yes, little Shah Rukh Khan, what is going on? Yes. Who is he? Who is he? Okay. Is this whole thing clear? Tell me. Do you wish to watch the video again or have any doubts? Yes. Clear? Yes, Shillong. Itanagar, Chuachandpur. Shillong is very disturbed today. I don't know what. Why? Shillong is very disturbed today. Especially this section. This area on my left. This is very disturbed today. Especially the first bench. Oh, sorry, second bench. Yes. Don't be distracted. Please concentrate in your class. This is going to be important. This is actually... Yeah, I know you want to watch it again. That's why you were behaving like Shah Rukh Khan, I know. Okay, fine. No issues. So this is clear. Let me ask you what is rigor mortis first. Then only I will be showing you video. What is rigor mortis? I have told you that before video also and after video also. What is rigor mortis? Yes, tell me what is rigor mortis. All of you. Itanagar, Shillong, Chuachandpur. Then only I am going to show you the video. Tell me what is rigor mortis. Yes. Yeah, it is stiffening of body, very good. And the reason being, Itanagar, Chuachandpur, Shillong, what is the reason of stiffening? The reason is lack of ATP. So if there is no ATP, what is going to happen? the relaxation process is going to not take place. So the muscle will remain in permanent contraction position, contracted, and that is what is called as rigor mortis. So this happens to a dead body. So dead bodies, they show this particular kind of thing. Okay, so you are asking for the video once again. So let us see that.
This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not you use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T-tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Okay, children, got an idea? Yes, got some idea? Shillong, Itanagar, Chuachandpur, something is clear? Okay, fine, very good. I'll be sending you this video on WhatsApp. You can see it in a never-ending mode. Okay, fine. So, let us first of all draw a stick diagram, what is happening. So, this was my actin, and in between my actin I have what? I have my myosin, the red thing. So, now what happens is, the whole structure starts to the myosin will start to pull. This is what is called as sliding filament theory. The myosin starts to pull it in a way that the distance between the actin starts to get shorter and shorter. And there is very less region. The myosin will remain at its original position, but yes, there would be, I said today is not my day for drawing. I don't know what is happening. Okay. So this whole thing is now represented as this thing. So this is sliding. What is sliding? The myosin always remains fixed, but yes, actin slides over myosin. This is sliding filament theory. So myosin is always static. What is dynamic? Dynamic is actin. So what happens is, can you see the change? This was the region, this was the sarcomere thing, and the sarcomere thing has contracted to an extent that it has become so short. Yes. Can you see this? Okay, fine. Shillong, do you want me to call Ashish sir? What is going on? Yes, tell me why you people are so reluctant today. Shall I call Ashish sir? Do you want me to do that? Isn't it? Please behave yourself in class. You people are grown-ups. Don't behave like children. Don't need a supervision in your class. Do you? I don't think so you need. Okay, please behave yourself, everybody. It's a request. So this myosin is actually static, and what is changing its position? Yes, the actin is going to slide over it in a way that whole sarcomere becomes smaller and smaller in size. So let's see what is going on over here. So first of all, let's mark with every distance, mark with every thing. So sarcomere, its length decreases. This is what is decreases. Okay, sarcomere's length decreases. What about A band? Decreases or increases? Tell me. What about I band? Just analyze your copy and tell me. What about I band? What is it doing? Decreasing or increasing? And what about H zone? Tell me the values of these four things. What are happening with them? Yes. All these four things. All of you. Tell me what is going to happen with sarcomere. Sarcomere thing is very clear. See. Sarcomere was this long earlier. It was to be this long earlier. Now it is shorter in its length. It has gone half. It's just half in its length now. So its distance has, its length has just been halved. Okay. Now what we can see over here, A band. So let's see quickly what is A band. So A band was what? A band was the overlapping region, isn't it? So A band was the overlapping region, and basically the A band was described by the myosin. So the myosin is present over here also, over here also. So this is what is the A band region. Let me just draw it in yellow. So this was the A band region. This thing was A band. But yes, myosin remains constant. Myosin has not changed its length. So obviously, excuse me, this A band region is going to have same, no changes. What about I band? Yes, I band, let's see this red section. We just remove this yellow part, be able to show you nicely. So this red section has actually penetrated inside the areas, and this is somewhat like this diagram. This should be this way. So we can see what was I band. This particular yellow thing was I band. If you just this particular yellow thing was I band, isn't it? So we can see over here, yes, the I band has decreased in its length. You can see here over here. So I band has decreased in its length. And what about H zone? Yes, you can see H zone. This was the particular thing, H zone. Let me mark it in different color. Let's see in green. So this was the region of H zone, this green thing, where there was only myosin but not actin. So this region, the green region, has also decreased. Can you see? Because the actin is actually getting into the H zone, none other than H zone. So H zone region is also decreasing. So this is the kind of question you are expected to be asked in NEET. Okay, this is the kind of question you are being asked in NEET. So this is quite important. You have to keep these kinds of things in mind. That sarcomere, yes, it decreases. A band, yes, it remains same. I band decreases, and H zone also decreases. Why H zone decreases? Because yes, the particular insertion of the actin will be none other than in the region of the H zone. So this is how the whole process of the contraction of muscles is taking place in your body. Okay. The sliding filament theory was proposed by two people together. They are said to be Huxley and Hansen. This was sliding filament theory in 1954. Yes. Let me know if anything is not clear. Yes. Are we clear up to this? Is everything clear up to this? Tell me yes or no. Shillong, Itanagar, Chuachandpur. Is everything clear? Okay, fine. Very good, Itanagar. What about Shillong? Yes, Shillong, is it clear? Okay. Chuachandpur? Tell me, children, if it is clear. So this was all about your sliding filament theory. This was about the contraction section. Let's move ahead to the disorders part. So when you talk about the muscle disorders, we all know muscle and bones, since we have studied this whole as a single unit. So we'll be seeing the disorders also as a single unit. Okay, do you want anything to be revised or repeated? Tell me. Yes. Yes, Shillong? Clear? Everything is clear? Okay, no revision, nothing? Okay, great. Just let me know in case you need to have anything. Okay, fine. So what we have now is, how many of you have heard about, what we say is muscle fatigue? How many of you have heard about muscle fatigue? So there is like 3 categories of muscles: the skeletal muscle or the striated muscles, the non-striated or smooth muscles, and we have the third category, that is cardiac muscles. So skeletal muscles, yes, they show muscle fatigue. Non-striated or smooth, they never, and the cardiac also never shows muscle fatigue. So you have a special category of muscles that are showing this kind of movement, this kind of thing. They are said to be what? They are said to be skeletal muscles. And why do they show this thing? The skeletal muscles show fatigue. When they produce what? Lactic acid. And when do they produce lactic acid? When they produce lactic acid, when they are performing anaerobic respiration. So whenever they are doing anaerobic respiration, they are out to produce this lactic acid. So what happens to the lactic acid they are producing? Yes, that must be actually because of product of anaerobic respiration. So this lactic acid is actually, the lactic acid enters the liver, and liver we have a cycle called as Cori's cycle. What happens in this cycle? We have the lactic acid getting converted into glycogen and CO2. And this CO2 is again, this is somewhat the 1/5 of the part, sorry. This is going to be the 1/5 is turning into CO2, and 80%, that is 4/5 of the lactic acid converts into glycogen, and this glycogen again is sent to muscle. So this forms muscle glycogen. So this is how your liver is actually working hard in reviving back the lost glycogen in the form of lactic acid. So this is the particular thing called as muscle fatigue. So in absence of oxygen, all these things, they happen with our muscle. When there is no or there is absence of oxygen. Okay, so moving ahead, what we have next is, disorders. So muscle fatigue is not a disorder. We will see one disorder that is called as hypertrophy. So hypertrophy is increase in muscle mass. So whenever there is increase in muscle mass, we call it to be hypertrophy. And then we have is atrophy. Now by now we know means absent in biology. So this is obviously a decrease in muscle mass. So increase in muscle mass is said to be hypertrophy. Decrease in muscle mass is said to be atrophy. Apart from this, we have a very famous disorder called as arthritis. So what is arthritis? Anything that leads to malfunctioning of the joints. So whenever there is malfunctioning of joints, is said to be arthritis. So for arthritis, you have one more disorder called as rheumatoid arthritis. So what is rheumatoid arthritis? This is actually the most common inflammation of synovial joints where too much fluid is produced and because of this too much fluid, what happens is the joints swell and become extremely painful. So this is the whole scenario how the rheumatoid arthritis is happening in a system. Okay, just let me know if anything is not clear. Yes, children, just let me know that. Just let me know if anything is not clear up to this. Shillong, Itanagar, Churachandpur. Yes, children? Clear? Okay, very good. Shillong, Churachandpur, Itanagar. Shillong, just give me a thumbs up if it's clear up to this. Yes. Okay, very good. Thank you so much. So, I'm moving ahead with next disease. This was up to rheumatoid arthritis. So, it's an autoimmune disorder. How many of us know that? Rheumatoid arthritis, it's an autoimmune disorder. How many of you know this thing? You must have studied this in 12th also. It's an example of autoimmune disorder. So, autoimmune disorder is a disease where the own cells of our body start to face problems from the immune system. Okay, let's now see the next thing. It's osteoarthritis. Osteoarthritis. So what is osteoarthritis? This affects only cartilage. So if you talk about only cartilage, where? At synovial joints. So this is going to affect only at cartilages at synovial joints. So what is going to happen? Tell me. Yes, the cartilage erodes or it gets wiped off. Erodes from there, from the joints, and they need replacement, isn't it? They need to be replaced, but the problem arises and are replaced by bones. So what happens is there are deposits of bones at places. So obviously this is going to cause bones in lumps. So this obviously is a painful situation where the bones, they come and they have a great friction with each other. So because of this friction there is arthritis. So always you remember, arthritis is something which is, this means something which is related to inflammation. And I have taught you already that inflammation has 4 signs, that is redness, rubor, tumor, calor, dolor. Remember? So rumor was redness, tremor was swelling, calor was heat, and dolor was pain. So when these four things they come together, wherever they appear together in a person, the person is said to be affected from inflammation. And when we talk about osteoarthritis, osteo means bones, arthritis means painful joint. So obviously the bones are the region, the deposition of the extra bones in the place of the cartilage is the reason behind osteoarthritis. So let's come to next. This is one again. Once again we have done this disease, gout in excretion part also. Let's do it once again. So gout, what is gout basically? There is deposition of uric acid crystals. There is deposition of uric acid crystals. And what are these uric acid crystals going to do? What are they going to do? Tell me. Obviously, is going to make the joints become painful, and what else can happen? Swollen. So they swell and they become painful. This is gout. Okay. Next is like osteomalacia and rickets. So osteomalacia, we have been studying these rickets and all. Deficiency of, isn't it? Deficiency of calcium, potassium, sorry, phosphorus. Deficiency of calcium plus phosphorus. So if it in adults, if it is in adults, we call it to be osteomalacia, and in children it is said to be rickets. You remember the bow-shaped or the pigeon chest characters. The rickets are having characteristic that particular features. If a question comes to you because of rickets, both of these situations, the above situations, they actually make the legs bend. In these both conditions, the legs will start to bend to form a bow-shaped structure, somewhat like this. So it's the foot part. So this is somewhat like this. The patient has somewhat bow-shaped legs because the particular bones are not that strong that they can bear the weight of the body. So what starts to happen is that the body starts to put pressure, and the person starts to have a bending in the legs. So this is what is called as osteomalacia. Okay, one more thing that came like previous year paper that was in it was tetanus. So tetanus is what? Tetanus is always defined by sustained contraction. Initiated by impulses from many muscle fibers. So there are many muscle fibers which actually start this particular contraction. They initiate these contractions, and once the contraction has been initiated, what happens is they enter into tetanus. So this was a question last year. What is tetanus? So the answer was spasm is also called as tetanus. When talk about spasm, this is also a form of tetanus only. So spasm is tetanus. Okay, there is something called a strain. Strain and sprain are different things. They are not the same. Strain and sprain are different things. So I am giving you a statement, just rectify me. I am just giving you a statement. The statement is that the length of actin and myosin do not change during contraction. So tell me if this is wrong or right, true or false. The statement is this. This happens to be a statement. Tell me if it is correct or incorrect. Yes, children. Oh. Yeah, quick. Length of actin and myosin do not change during contraction. True or false? Itanagar, Chuachandpur. The chapter is done now. I'll be just asking you small questions. Chapter is done from my side. Okay, so we'll be starting with a new chapter tomorrow. That would be, I guess we are talking about central nervous system, exactly. So we'll talk about central nervous system tomorrow. We'll start with that chapter. Yes. Tell me, answer me this question fast. Itanagar, Chuachandpur, Shillong. What is taking you so long? Is either true or false? Yes, Shillong? Yes. Shillong is yet to answer. Okay, fine. They are showing me. Okay, this is false. Why? Show me how the actin and myosin is changing. Tell me. See this diagram. This is the actin part. Okay, this was the original scenario. Suppose, and this was the myosin part. Okay, so it starts to contract. What would happen? See, has it changed? See, is it changing? Tell me if it is. The myosin is going to get inserted in this section. No, it is not. So actin and myosin, they don't change their sizes. It actually, the actin starts to slide over myosin. So sliding never means changes. Okay. Actin and myosin, they don't change their size, but they actually overlap the H zone. The H region is overlapped and the actin gets a chance to enter inside the H region. So this statement is very, very true. Okay, don't get confused. Okay. Okay, now you tell me any doubts? Itanagar, Chuachandpur, Shillong, do you have any doubts? And what about sarcomere? It increases or decreases? The sarcomere length increases or decreases in the whole process? What about sarcomere length? What about sarcomere in this whole process? Yes, children, tell me. Sarcomere increases or decreases in the whole process? Chuachandpur, Shillong, Itanagar. Tell me, sarcomere increases or decreases? Itanagar. Just an E from Shillong. Okay, fine. Itanagar, tell me. Chuachandpur, tell me. Increases or decreases? Yes. Obviously, sarcomere is decreasing. You can see this image, the diagram also. It always decreases. What about A band? decreases or increases? A band decreases or increases? People are ready with bags, huh? Shall I also go and get mine? Shall I also should go and get mine bag, isn't it? Yes. Obviously no change. And what about I band? What about I band? Tell me. Yes, I band decreases. Very good. Very good, Itanagar. Keep it up. Okay, nice, Shillong. And what about H zone? What about H zone? Decreases or increases? Yes. Yes. So this was all about this chapter. Go back home and study this whole of the things like together once again, so that you don't have any doubts. Let me see what is the syllabus for your test. We'll ask somebody to update it by today evening, so that you can have. Okay, breathing exchange part was done totally. Breathing exchange part was done by all of you. Breathing exchange. I gave you an assignment. Did you match your answers? Shillong, Itanagar, Chuachandpur. Don't leave the class before I ask you to. Yes, answer me my questions first. Okay, fine. It was done. All of you. Did you match your answer? You are satisfied with your performance? Okay, fine. Very good. Okay, fine. So let's wrap the class. Today we are having a bit early wrap up. Let's see. Tomorrow we will be extending 10 minutes more. I will manage this 10 minutes in tomorrow's class. Okay, fine. So let's go back home. Like always, start revising, and if it's your test, please study very, very hard, very, very nicely for your test, because all this hard work is going to pay you back in future. Okay, you need to be serious about all these things. Let's meet tomorrow again, and we'll start with central nervous system section. And obviously, go back home, have a good day, have good food. Okay, thank you so much. Okay, bye-bye. And be a little mischievous. Be a little mischievous. Okay? Okay, fine. Bye. Bye-bye.